Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we jump into this new episode, we'd like to take a moment and thank you for your continued support. Many of you asked how you can support us. One great way is to subscribe to the Dr. GPCR newsletter. Each month, we collect and curate information about the latest scientific advances in the field, as well as biotech and pharmaceutical news. Subscribe today at drgpcr.com/newsletter. We would also like to hear from you. Please take a moment to tell us how to make Dr. GPCR work for you by filling out any of our surveys on drgpcr.com/survey. You can tell us what you think about the podcast, the summit, and even Dr. GPCR in general. Stay tuned as we are working on the 2022 Dr. GPCR Summit edition. And we also have been busy working on a brand new secret project. Visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. I'm so excited today to have with me Dr. Pina Cardarelli as my podcast guest. And as you can see, if you're listening to the podcast, you cannot see, but when the video gets up on YouTube, you will be able to see that the typical background that I have is not the same. We're out of our typical location, but nevertheless, super excited to have our guest with us today. Hi, Pina.
Hi.
It's so great to have you on today. I'm so excited.
Thank you. It's a pleasure to be here.
Thank you. Thank you. I really also loved your presentation at the Dr. GPCR Summit last September.
Okay, that's sweet. Thanks. Thank you.
It's really interesting. So let's start off with my typical questions. Can you please give us an introduction as to who you are today and kind of a historic rundown as to how you got to where you are today?
Oh, great. So obviously, my name is Pina Cardarelli, and I am currently the CSO at GPCR Therapeutics. And additionally, I am president of a recently established site in the San Francisco Bay Area, which I'll talk a little bit more about later. As far as my story, I do think it's quite an interesting story. And I think what's important here is for you to remember that as a woman scientist back then, we were not well represented. So I was typically the only woman in the room. So that, I think, really shaped me. Additionally, I came from a poor immigrant family that moved to the United States from Italy. And so I came to this country, obviously not able to speak English, but I did have several role models that I think really shaped me. My first role model was my fifth grade teacher, and she loved to travel, and she taught me to really aim high and travel. So I still, to this day, I really love traveling the world. My second role model was my high school biology teacher, who really said, introduced me to human biology, and I was fascinated, and that led me to pursue biology in college. And then my third role model was my genetics professor in college. And he did something quite interesting. He really instilled confidence in me to consider graduate school rather than what I was thinking as going into a hospital and working in a lab. But he really gave me the confidence to really consider graduate school. So I went to Albany Medical College, where I got my PhD, and it was in a field called physiology, which is not a common major nowadays, but essentially it's pharmacology was what I was working on. And so that was up to my PhD. And then I made a bold decision to travel from New York to San Diego. And I took on a fellowship at the Burnham Institute in San Diego. And it was an exciting time because they had just purified the first integrin cell adhesion receptors. And I was there during that very, very exciting time. And I also thought, okay, I'll stay here for a few years and then move back to New York. But I loved San Diego. And so I took on a position to work on a small molecule integrin antagonist at a company in San Diego. But career-wise, my big move came when I was recruited to the Bay Area. So I was recruited by a company called Coulter Pharmaceuticals, and that's when I began my journey into biologics. Really, for most of my career, I've worked on biologics and less so on small molecules. And there, I had the task of trying to understand the mechanism of two CD20 antibodies, Rituxan, which was our competitor, and Bexar, which is what Coulter had, which was an iodinated CD20 antibody. And I was really fascinated with the antibody therapeutics, how antibodies to the same target could differ. And that really engaged me. However, Coulter was acquired by Corixa, and then several programs and personnel, including myself, were acquired by a company called Metarex. So here I went for 3 years, 3 different companies. It was quite a shock, I must say. But anyway, I joined Metarex, and I'm sure some of your listeners are familiar with Metarex. I'll talk a little bit about that company. I loved working at Metarex. It had the right number of people, the talent, the culture. My boss was absolutely brilliant, and he always supported new ideas, and he also understood me well, so it really made for a great relationship. Now, it was at Metarex that 2 checkpoint inhibitors were identified and antibodies were generated, CTLA4 or Yervoy and PD-1 nivolumab. And my group worked on establishing many of the in vivo studies to advance these programs. So I was intimately involved in IO at a very early stage when people were very skeptical, basically. In addition, at Metarex, I also worked on targeted oncology, number of targets, CD30, CXCR4, which I'll talk a little bit more about, fucosal GM1. I worked on interferon receptor 1, interferon alpha. I also worked on antibody drug conjugates targeting CD70, mesothelin, glypican. So I had quite a diverse background. And again, everything was centered around biologics. But then Metarex was acquired by Bristol Myers Squibb. And what Bristol Myers Squibb did is, because they came really well funded, a lot of these programs that were just percolating along were entered into the clinic. So in that part of my career, I learned so much about later stage drug development, not just the discovery part, but what it takes to file an IND and get it into the clinic. So that was a great learning experience. But then in 2018, there was a reorg at Bristol Myers Squibb, and I left and I started to consult for a number of companies. And I was approached by GPCR Therapeutics to consult. And I started with 10 hours a week, and rapidly it became full-time. The CEO of the company has just really given me a lot of support to, advance his vision, his company vision. The culture there is very collaborative. People are easy to work with. And their technology, as I mentioned, when I did the podcast, was GPCRs form heteromers, and in doing so can induce enhanced signaling when both ligands are present. So the idea is to do dual inhibitors to block the signaling. And the company focused on CXCR4 and found that CXCR4 can form heteromers with 42 other GPCRs. And so that is why they were interested in me as a consultant, because of my experience with not necessarily GPCRs, but specifically CXCR4. So at GPCR Therapeutics, we have acquired a small molecule CXCR4 inhibitor, and we're in the process of bringing it back into the clinic, into a phase 2A trial for stem cell mobilization. And this is really exciting for me because, like I said, I really do now enjoy the later stage of the drug discovery process of really bringing things into the clinic. So I have found that this is a great learning experience. And as I mentioned, we opened up a U.S. site in the San Francisco Bay Area, and we are currently 7 people, and we hope to really grow. So there's a Korean site with about 30 people, and there's 7 people here, and we're hoping to grow in the next year. So that is pretty much my background. I know it's a long story, but I felt it was important to really reflect on many aspects that have really influenced me in my career.
Thank you for that. I looked you up on LinkedIn, and having the context of all those positions that are listed on LinkedIn is just priceless. And I really appreciate you telling the story. But I would like to take a step back to the 5th grade, or maybe between the 5th grade and high school. Were you always interested in science and biology?
No, I don't think the biology came to me when like I said, when I was in 10th grade, and I took a biology class, and then especially when we got into human biology, I was very fascinated, and it was at that point that I realized that I really did enjoy the science, and it was intriguing to me. I've always been more of a quiet person, I think. I think of myself as relatively quiet and more introverted, and a love of science and data. So,
and as a child, when you think back, what did you want to become when you grew up?
So when I was young, I wanted to become a school teacher. And I think that was because my first role model was, in fact, a school teacher. And so that to me was the opportunity for women back then. You didn't have what women have now, which is great, of course, but you had much more limited opportunities or role models, if you will.
Thank you for that. And I really enjoyed how you really mentioned the fact that when you started out as a scientist, the opportunities were small, or were more difficult to come by, and also you were oftentimes the only woman in the room. So, 2 questions out of there. What did you learn as being the only woman, and how did you overcome, these difficulties?
I think I learned to to speak out, but in a very quiet way. And I learned that what really was going to help me to progress is data. I'm very, very strict with my data. I feel that what really needs to guide us in this profession is really strong data, strong experimental results that really lead to a decision, and that you're progressing down the right path. So I think it was the data that gained respect for me in my career.
That's fantastic. And I think you're correct. Having the data speaks for itself. You have the correct data, you have the right controls. then there's no argument there. The data is the data.
Mm-hmm.
You started out at GPCR Therapeutics on a consulting basis, and then now you were hired. You have all this experience, but this is the Dr. GPCR podcast. So my next question is, how did you get into studying GPCRs, and when and where did you acquire your expertise in CXCR4?
Okay, so I think because there's been so much data surrounding CXCR4 pathway in cancer, we at Metarex developed an antibody to both the ligand CXCL12 as well as the receptor CXCR4. So that is really when I became interested in GPCRs. And we did a lot of in vivo studies in xenograft models comparing the two targets, so antibodies to the ligand and the receptor. And we were really impressed with the efficacy of ulocuplimab, which targets the receptor, in a variety of heme models. And I was involved in developing ulocuplimab from, starting the project, testing the antibodies, all the way to bringing it to the clinic. And people laugh at me, but I still call Ulo my baby because I was just so much involved in it. And now working on CXCR4 again at GPCR Therapeutics is really, really exciting for me.
It's really bringing you back home.
Yes, my baby.
Lovely. Well, actually, the first GPCR I worked on ever was CXCR4. So it does have a special place in my heart. I started out as a summer student in Nicolas Devecker's lab at the University of Montreal. And I had to make 6 mutants, 3 in the dry motif and 3 in the N119 area of CXCR4. And back in the day, we were using these single-stranded DNA phage methodology to make the mutants, so no PCR. And I think it took me, as a summer student, the entire summer just to make the mutants. And when I did my thesis, my master's thesis, we ended up publishing the results of this data showing Brett measurements with these mutants, comparing them with the wild type. So we looked at GTP gamma S signaling and GTP gamma S binding, thanks to some collaborators, but also did all the Brett experiments when it came to homodimers of these mutations. So, and it was it was an express publication with, I think, 2 or 3 figures, but the background work that went on and the amount of data I had to generate, this was my first paper, was huge. But CXCR4 always has a sweet place in my heart.
So it's your baby too.
It is. Yeah. And the next one was CCR2, and I saw on your LinkedIn that you at some point worked on MCP1, which is one of the ligands of CCR2. Yeah. Which was a much, I want to say, easier project to work on. The data was much easier to acquire, but chemokine receptors in general have a special place in my heart.
Yeah, right. I agree.
I think they're just so fascinating to be working with.
Right. But it's interesting that there's not, and maybe I can talk about it a little later, but there's not many FDA approved drugs, antibodies to GPCRs, specifically chemokine receptors, other than CCR4. So it needs some more work, basically, to bring it to the next step.
Agreed. Do you think this lack of FDA-approved antibodies or even small molecules targeting chemokine receptors is due to the fact that we don't fully understand their biology?
I have a lot of thoughts on that, and I think that part of it is just generating the reagents to make useful antibodies. to GPCRs, I think is still a big challenge. We did a lot of work to generate the antibody to ulocuplumab, and at the end we only had like 3 antibodies that looked promising. Whereas, when I worked on other targets, let's say mesothelin, you have hundreds of antibodies that you can choose from that bind to different epitopes and to different functions. So I think that there is the challenge of getting enough of these reagents to really generate a good panel of antibodies for selection.
Sounds to me that technologically speaking, there's a lot of advances that need to be made in order for us to get to that point where I won't say it like this, but we can generate antibodies faster and have a larger final pool of potential targets.
Yeah.
But we love our GPCR, so we're not going to give up.
Okay.
So as I mentioned, one of my questions is also about your experience as a CSO of a successful company. How does your day-to-day look like? How is it different from your previous role? What are your thoughts on being in that C-suite level position?
Well, one of the things for sure is that I went from a very, very, large company, essentially at the end, and even Metarex had like 450 people. So going from larger companies to a small company is a big challenge because, for example, if I needed a specific assay, I would send, request it from another group. Or if I needed any advice, I would just go to a colleague and, what do you think of this data? It was just very, very easy. But this type of specific expertise, is much more challenging with a small company. So how I've selected to overcome this is to bring in some outstanding consultants, especially because now we're bringing something back into the clinic, and GPCR Therapeutics doesn't really have clinical experience. So having these expert consultants with so many years of industry experience has been absolutely essential. So I try to overcome those limitations by bringing in the experts and experts in other areas as well, not just the clinical aspects. I think the other challenge that I faced is the language barrier. I do not speak Korean, and also there's a big time zone difference. So meetings are often later in the day for me. So I think those are 2 challenges. But I have to say that I'm really impressed. Since 2019, when I joined, I've seen a lot of progress in their English speaking skills, because we have meetings and they have to present their data and they have to present it in English. So I've seen great improvement there. So I think it's working out. I'm finding ways to work around the challenges, basically.
And the good thing is that no matter who your colleagues are within the company, the data is the data and the science remains the same.
Universal language, right?
Yes.
When you look at those dose response curves.
Those still give me goosebumps. Every time I see a beautiful dose response curve, I'm like, oof, so nice. Especially if you've done the experiment multiple times, if you're doing them, yourself, and then you plot the data and it's late at night and you're looking at the data and it's just beautiful. It just gives me personally that satisfaction of a job well done, kind of an artistic type of satisfaction.
Right. Emails at night are always welcomed if they have some very nice data associated with it.
Yes. Agreed. So I know we kind of talked about this, and we all agree in general in the GPCR community that GPCRs are still good drug targets. But in your own words, what is your thought about GPCRs being good drug targets?
So I think that there's such a large family of receptors that I think that we haven't really tapped in and fully understood its potential. And one of the things, and I brought this up a little bit earlier, is an area I'm interested in is the antibody. So many of the molecules right now targeting GPCRs are the small molecules, which is obviously interesting, but I'd like to extend and go beyond the small molecules in the future. The antibody global market is like 210 billion. So it's really a good lucrative area to be in. And as I mentioned, there's two approved GPCRs that are biologics. There's 70, I believe, in clinical trials, but only 2 approved. And one is the CCR4, which is the enhanced ADCC antibody for cutaneous T lymphomas. So I feel like there's a lot of opportunity, and that's where I think we could potentially go. I think orphan receptors, there's still a lot of opportunities there as well. And I believe, from my perspective, focused on heteromers, that perhaps we can bring in, bias signaling or allosteric modulators, that could potentially form new targets and increase the availability of GPCR targets. So that is, bias signaling, new signaling pathways, or changes in potency. I think one of the things that we are working on is the heteromer hypothesis, and perhaps we can make some progress there. And then I think one area that I'm very interested in is drug repurposing. I think that this has a lot, a significant amount of pros. So cost effective, time efficiency, because you don't have to conduct as many, trials. They come well understood with respect to safety, so you know what the toxicity profile looks like. And I do feel that we are obviously working on CXCR4 in combination with adrenergic beta 2 receptor antagonist propranolol. Again, that is a repurposed drug that we're hoping to bring back into the clinic for stem cell mobilization. But there are many other examples. So, for example, thalidomide has been repurposed as Revlimid for multiple myeloma. So there's opportunities there, many repurposed drugs. So I think spending some time working on repurposing some of these drugs, I think, would give us some leverage. And of course, what we hope to do is, if we get proof of concept with 2 individual drugs, for example, then the idea would be to either make one molecule that can do both, or improve on the propranolol, for example. So this is just, I just do think repurposing drugs is a good way for us to go.
I love the idea of repurposing drugs for the same reasons that you just mentioned. Is really, you have so much data already on that drug. And then the toxicity questions are, for most part, really well answered, and then you can build on that. I had maybe a follow-up question regarding repurposing in the context of CXCR4 and beta adrenergic. You had mentioned that the goal is to mobilize stem cells.
That's correct.
In what context? Because if I understand correctly, there is the Mozobil, the EMD 3100, that already is used in the clinic to mobilize stem cells. Would you be targeting different cells, or in a different context? What does that look like?
Yeah, very good question. So the goal is to combine it with propranolol, with the idea that propranolol improves the bone marrow cellularity, and therefore, when we do the mobilization, we would be able to mobilize cells that have the potential to form a different immune cells. And so the idea is that we would also try to remove or eliminate GCSF. So the current approved therapy is definitely using Burixafor or Mosavil in combination with GCSF. So that is the standard of care. Our idea is to determine if we can eliminate the need for GCSF by using the propranolol, which hopefully will enhance this bone marrow cellularity and mobilize more active cells, essentially. So the one other thing I wanted to mention is, GCSF requires injections, requires 4 to 6 days of therapy. Patients have to go in. Most patients are unable to do this on their own, so they have to go in for consecutive days. Now, if you turn that around and take an oral drug like propranolol for 7 days, let's say, they can take it at home and then just come in that day for the mobilization. I do think it will provide patients an opportunity to really reduce the amount of stress that's associated with these treatments.
I love the idea, and yes, now that you're mentioning it, I do remember that Mosavil is co-administered with the GCSF, and then you have to be there, and it takes quite some time. And agreed, it seems to be a much simpler option of taking a pill, preparing the mobilization, and then going in on the day of, get what you need, and then moving on basically from there.
Right. Yeah. So that's the hope. And as I mentioned, we're going to go back into the clinic, hopefully the middle of next year. The fact that we were able to acquire this drug that had been in the clinic and we're now just resynthesizing it makes it a lot easier, because it's already, it's got an IND and everything is much faster because of it.
Lovely. Let's take a little bit of a zoom out. And what are your thoughts about the most interesting new developments in GPCR drug discovery?
Okay, so I think, for me, I would say that the information, the structural information that's now becoming available is quite interesting, and I think that, I guess there's quite a few things to think about with respect to what is taking place. But I think the structural information is going to help us. Ability to actually purify more easily these molecules, I think, is an important thing as well. So I would say that there's a lot happening. The structural revolution. And understanding the activation and inactivation, that's another area of interest.
I also love the idea of leveraging heterodimers, which is not something that a lot of people or a lot of companies have been thinking about in the context of a clinical application. And I think that's really a nice way also to approach the question. And I don't know if you've seen the article about the CXCR4 tetramer. I have to bring it up. But what are your thoughts on recent cryo-EM structure of the CXCR4 tetramer? So what are your thoughts on leveraging more than a dimer in the context of a specific disease? Could we be thinking about, and I'm completely fantasizing here, but could we be thinking about molecules that bind 2 or 3 units out of a tetramer in the context of a disease?
That is a fascinating idea, and I can think of several, targets for which that would be applicable. If you think about targeting both the cancer as well as the immune component, having a complex molecule that can work. Obviously, this would have to work in trans, but I could see some utilities there as well.
So we've talked a little bit about the key challenges in GPCR drug discovery when it comes to being able to get more structures, to being able to purify the receptors, obviously to get more structural information. And we've talked about the idea of targeting GPCRs in IO in the immuno-oncology context. What are some of the other disease areas that have connections to GPCRs where we should be focusing or we're not focusing as much as we should?
Yeah. So the area that really interests me, although GPCR Therapeutics is not working on this, is in the area of neurobiology, with the fact that the aging population in the US. I believe that developing GPCRs that can target like Alzheimer's or Parkinson's, for example, is an area that needs new drugs. There's a lot of drugs, but the failure rate is really pretty high. I believe that there needs to be a lot more effort to really improve the success rate there. And there was a review by David Gloriam who highlighted this, Alzheimer's, being a good area to consider. And so I think, besides the others that we discussed, I do believe that, considering Alzheimer's diseases or Parkinson's, for example, is really a good place to be. Of course, in oncology, we're focused on oncology. We're now working with Dr. Paul Insel, in the area of oncology. And then I know many companies are working on immuno-oncology. And here's a great opportunity because, as you know, tumors have many ways that they evade the immune responses. So thinking about GPCRs that target MDSCs or Tregs or M2 macrophages or TAMs or exhausted CD8 T cells, I do think it's really important. And it's very clear that the stress response is associated with cancer progression, and it has a big impact on immune cells. So I believe that other companies will probably make progress here. I assume many companies, I know many companies are working on this. So I do think that that's another area where we will begin to see more development.
I think there's a lot of work to be done in the field, and especially when it comes to not only understanding how GPCRs function, whether they're alone or in dimers or tetramers, but also how we can target them better for different diseases. What do you think are the key tools that we still need to develop in order to speed up GPCR drug discovery? We've talked a little bit about cryo-EM or structures, purifying receptors faster and better. What else do you think there is?
When I joined GPCR Therapeutics, because they have, like I said, 42 other GPCRs that associate with CXCR4, and then they have a large panel of over 1,000 other interactors that need to be evaluated. When we started thinking about which ones and trying to determine which one would be the best next GPCR to work on. What we realized was that there's a big void with respect to validated reagents. So that includes, antibodies, so you can determine the expression, the co-expression, for example, on the cell surface. Whether you're using flow or IHC or proximity ligation assay, there's just very limited tools. So I think that's one of the areas of limitation. The other is, and my group is probably tired of me saying this, I believe there should be more focus on endogenously expressed cells. I'm worried that transfected cells and overexpressing cells in the GPCR area can be a little bit misleading. And yes, let's get your initial observation with those transfectants, but then let's bring it back to what I call reality. And I really like to see endogenous. But again, when you don't have the validated reagents, I think it really is a bit of a setback. The other is RNA. There's been a lot of it's a good first step, but I do think that validating it as as protein on the cell surface is important. The other limitation, and this is very close and dear to my heart, is the mouse models. I think that, the availability of proper mouse models is limiting. I think part of it is the lack of cross-reactivity. Even I thought, okay, when you go switch to small molecules, they're going to be cross-reactive and have the similar affinity between the human and the mouse. But that's not always true. So there's that issue of cross-reactivities, whether you're talking about small molecules or antibodies. And then importantly, the antigen may be expressed in certain tissue or organs in the human and not in the mouse. So I think you have that challenge as well. And I do think, mouse models with human immune cells helps a little bit, but there still could be differences. So I think the challenge of the right animal model is really critical here.
I love that. Yes, I was listening to you talking, and in my head, I was thinking, you need to have the correct model. It's like, choosing ice cream versus soy ice cream. It's not the same thing. It's called ice cream, but it's not the same thing.
Not the same, right? Right.
And I also loved your comment about going for the endogenous levels of expression and detecting the protein at the cell surface on endogenous tissue and cells. And that is something that's proven difficult with GPCRs. Again, going back to your comment about the fact that we don't have the correct antibodies, we don't have the tools to look at that. We talked a little bit about this in the beginning, about how you focused on getting consistently well-controlled, great data in order to establish yourself as a scientist and as a leader. If you had to give advice to junior or young scientists aspiring to contribute to the GPCR field, whether it's in academia or industry, what would that be?
Okay, so I actually love mentoring junior scientists. This is something I've always done and will continue. And the main advice I would give is to have confidence in yourself. I think that was a barrier for me. So have confidence in yourself. I think with confidence, you can, progress your career faster. And this is especially true for women. I see it more with women that I mentor than with men that I mentor. And I've already mentioned this before. let the data drive each of your decisions. Just learn from that data, and then always adhere to strict controlled study. I'm a very gentle, kind person, but if people forget those controls, you'll see a little bit of meanness come out of me. So that's important. And then the other thing I would advise is to communicate and collaborate with others, because you cannot do anything, you work alone. You really need to be really good at communicating, getting your ideas across, and working with people. I feel that is absolutely essential for their career.
That's fantastic advice. But I want to take you, you mentioned confidence and be confident. No one is born with confidence. Confidence gets built up over time. How did you build up your confidence? Or what were the determining moments in your life where you got to a point where you said, actually, I can do this because of past experiences?
I think it actually began probably actually a little bit later in the career than most people probably would understand. But I think for me, it came around the time when I started getting involved in biologics, and then went to Metarex, where I just felt that people respected what I had to say, respected my data. And I think that was really fulfilling for me, and realized, wait a minute, you can do this. Of course you can. Look at all these programs you're advancing. Look at how big your group has grown now in terms of direct reports, and how you're progressing up, from associate director, senior director, VP, so then I was like, okay, I can do this. But it did take a little bit longer for me than I'm hoping that people I coach can establish this much sooner.
In what context do you coach younger scientists?
Well, in previous, we had a mentorship program like at BMS or Metarex. Here now, it's mostly mentoring people who are in the organization who, I find GPCRs extremely complicated. I thought ADCs were complicated because you have an antibody, a cleavable, non-cleavable linker, and a payload. I thought they were complex, but GPCRs are very complicated. So I work with very, very, smart, know the GPCRs in and out. And I work with them by, saying, okay, that's great. Let's just step back. What's the bigger picture? What's the bigger question? So right now, my mentorship is mostly around people who work at GPCR Therapeutics.
Fantastic. And I love the idea of, pulling these very smart scientists back and giving them really the thought process of looking at the entire picture, and not only being focused on one or all those projects. I think that's a very difficult thing to do when you are in control of the data and you have that pressure of generating consistently reliable data, and then you kind of lose focus. And it's an art to be able to do both.
Right, absolutely. And the details are very important. I'm not trying to say that at all.
Of course.
I'm just saying that, that's how I help with the coaching.
And I'm sure it's very appreciated. We did talk at the beginning about the three people in your life: your fifth grade teacher, your biology teacher, and I can't remember the last person, who kind of shaped your university at the university advisor, who pushed you to go to graduate school, who shaped your career and propulsed you. But then the question I have for you is, what are the top 3 aha moments that you had as a scientist that actually did shape your trajectory?
Okay, that's a great question. So I'm going to start with ulacaplimab. Obviously, it's a CCR4 antibody, but it's an IgG4. So it doesn't have ADCC or CDC activity. And we put it into multiple xenograft models, as I mentioned, and it was really very impressive efficacy that we saw. And we were like, wow, how is this working? How is this working? And we started running a number of in vitro assays. And when we ran an apoptosis assay, and we saw that the antibody could induce apoptosis directly, and that this was also correlated with the expression. So rainbow cells, which have higher expression of CXCR4, had more induction of apoptosis, without cross-linking, just a direct binding. I think for me, that was like, this is what's unique about this molecule and how this could be working in our animal models, because it is an IgG4. So that was probably a big aha moment, discovering mechanistically the apoptosis. That would be my first one. My second aha moment was, as I mentioned, at Metarex, I was involved in running a lot of my group anyway, was involved in running a lot of the in vivo pharmacology studies with immune checkpoint inhibitors. And we were looking at anti-mouse PD-1, anti-mouse CTLA-4, and while each of them had some activity, when we did this experiment, when we combined the two, this was a real aha moment, because what we saw was not just additive activity, we saw true synergy, and we saw that we had cures in some of the mice. And that was like, everybody in the company was excited, and it was just a wonderful moment. The excitement was just so intense. And of course, this result was instrumental in, it was the basis for clinical trials now, where Opdivo and Yervoy are combined. So this really was a meaningful discovery, if you will. So I think that's probably one of my most important aha moments. And then you asked for a third. So the third, I don't know if it's an aha moment or more of a learning moment in my career. So I worked on antibodies to GPCRs, targeting both the ligand and the receptors. So, for example, I brought up CXCL12, CXCR4. I also worked on IP10 or CXCL10 and CXCR3. Additionally, from an immunology standpoint, I worked on interferon alpha and its receptor, IFNAR1. So, preclinically, antibodies to the receptor always showed superiority, but we didn't really know why, what it was all about. And then we out-licensed interferon alpha and an IFNAR1 antibody to AstraZeneca, and they actually did the clinical experiment. What they did is they brought to the clinic for lupus interferon alpha antibody, as well as interferon IFNAR1 antibody. And while both were efficacious, the activity or the efficacy of the IFNAR1 anifrolumab was superior, and so they decided to pursue that and now it's approved, FDA approved for lupus. And this was another very impactful occurrence that it just got approved. So it was a very exciting time for me to see that they did the clinical experiment that really showed that, at least in this case, that the receptor, an antibody to the receptor, gives you superiority. So I'd have to say those were three events in my life that were, very impactful.
Thank you. Thank you for sharing those. And it's so funny because I also worked on CXCR3. So you said IP10, and immediately in my mind, I was like, wait a minute, that's CXCR3's ligand. But before I let you go, I have a follow-up question regarding IP10 and the antibody detecting both IP10 and CXCR3. Why IP10 and why not ITAC?
So IP10 was started early on before I joined Metarex. And we decided that specifically for IP10, I don't know that we could have made a cross-reactive one, but I don't know what the rationale was for specific CXCL10 versus ITAC. I don't know that. What I do know is that we brought CXCL10 into the clinic and it had some activity, however, in rheumatoid arthritis. However, we decided to bring in a second generation antibody, which was more potent. And I think the failure for the ligand, and this is just my personal opinion, is that an antibody can prolong the half-life of the circulating molecule. So in some ways, it's almost like you have a circulating complex of CXCL10 with its antibody. And I don't think that's an efficient way to really, do what you need to do. And so by increasing the potency, we probably made that situation even more pronounced. So I'm more skeptical about making antibodies to the ligands than to the receptors. And this might be one reason.
Thank you for that. I am always fascinated to hear stories about chemokine receptors in general, GPCRs, but chemokine receptors and their ligands. Definitely. With this, Pina, thank you so much for taking the time.
You're welcome. You're welcome. It was a pleasure. I'm not an expert on GPCRs, but I am very passionate about developing drugs and bringing them into the clinic. So I do thank you for giving me the time to share my personal story.
Thank you so much for being here. And I want to tell our listeners that if they have a chance, they should go on drgpcr.com/summit-2021 to look up your talk that you gave at the summit. A fantastic talk about the work that you're doing currently at GPCR Therapeutics. And that reminds me, actually, you were just mentioning that you're building up the site and trying to fill it with a bunch of scientists. We do have on our career page some of these job descriptions. So I would like to invite everyone to join drgpcr.com/career and take a look at these positions. But also people can find you on our webpage, on the podcast page, and they can reach out to you and find out more about the positions that you have opened on site.
Yeah, we definitely have a position open for an MD to help us get into the clinic. So I will look and definitely put a post there.
Thank you so much, Pina. Take care. Thanks, everyone.
Thank you.
Bye-bye. Thank you for joining us for this Dr. GPCR podcast episode. We'd like to thank our guest, as well as our team members, Attila Forrest, Inez Pinero, and Alexa Truan. Please subscribe to the Dr. GPCR newsletter today and find us on YouTube as well. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.